the science behind PCR testing and genetic detection. Few scientific techniques have changed biology and medicine as much as PCR or polymerase chain reaction. Before PCR, detecting small amounts of genetic material was often slow, difficult, and sometimes impossible. Scientists needed enough DNA to study it, but many samples contained only tiny amounts. PCR changed that problem completely. Instead of searching for more DNA, researchers learned how to make copies of the DNA they already had. PCR does not create new genetic information. It simply makes millions or even billions of copies of specific genetic sequences. At its core, PCR is a method used to amplify DNA. Amplification means making many copies of a particular DNA segment. Imagine trying to read a single sentence written on a tiny piece of paper hidden inside a large, dimly lit room, but if that sentence could be copied millions of times and placed throughout the room, detecting it would be much easier. PCR works in a similar way. It takes a small amount of target DNA and repeatedly copies it for scientists to study. DNA contains the genetic instructions that help determine how living organisms develop and function. Every organism has unique DNA sequences, which makes DNA useful for identification and detection. Scientists can design PCR tests to search for very specific genetic sequences. If the target sequence exists, PCR can amplify it. If it is absent, amplification does not occur. This ability to target specific regions is why PCR is a powerful diagnostic tool. PCR relies on a repeating cycle of temperature changes. First, the DNA is heated so the two strands separate. This step is called DNA. Next, the temperature is lowered so short, DNA fragments called primers can attach. Finally, the temperature changes again, so a special enzyme called DNA polymerase can build new DNA strands. These three steps repeat over and over. Each cycle roughly doubles the amount of target DNA, creating exponential growth. Imagine making photocopies of a document. One copy becomes two. Two become four. After enough rounds, the number of copies increases dramatically. PCR works similarly, except it copies DNA. This comparison shows why PCR can detect tiny amounts of genetic material. A nearly invisible amount can become a detectable signal. Primers are one of the most important parts of PCR. They are short sequences of DNA designed to match specific regions. Without primers, DNA polymerase would not know where to begin copying. The specificity of PCR comes from primer design. Scientists can create primers that recognize particular genes, organisms, or mutations. Better primers mean more accurate detection. PCR became widely known during infectious disease testing because it can detect genetic material from viruses and bacteria. Even when the amount of genetic material is very small, amplification may allow detection. This makes PCR useful for identifying infections early. The technology became especially visible during the COVID-19 pandemic because PCR testing was used around the world. PCR looks directly for genetic material. Some rapid tests, by contrast, often detect proteins called antigens. Because PCR amplifies genetic material, it can often detect very small amounts that other methods may miss. However, this sensitivity also means PCR usually requires specialized equipment and laboratory procedures. Some viruses, including certain respiratory viruses, use RNA instead of DNA as their genetic material. Standard PCR works with DNA, so scientists must first convert RNA into DNA using an enzyme called reverse transcription. This process creates complementary DNA, or cDNA. The resulting DNA can then be amplified through PCR. This variation is called reverse transcription, PCR, or RT-PCR, and it became widely discussed during the pandemic. PCR is not limited to disease testing. Scientists use it in forensic investigations, environmental monitoring, and genetic research. DNA from crime scenes can be amplified. Researchers can study ancient DNA from fossils. Environmental scientists detect microorganisms. Farmers and researchers can identify genetic traits in plants and animals. This broad range of applications shows how versatile PCR has become. Because PCR is extremely sensitive, contamination can become a challenge. Tiny amounts of unwanted DNA may be amplified. That is why PCR testing requires careful handling, controlled environments, and quality assurance measures. If a method is powerful enough to detect tiny genetic traces, how do scientists ensure they are detecting the correct target? Much of the answer lies in strict laboratory protocols and validation procedures designed to reduce error. One of the most remarkable aspects of PCR is the speed of amplification. Each cycle roughly doubles the amount of target DNA. After dozens of cycles, an extremely small amount of DNA can become millions or billions of copies. That rapid multiplication is the reason PCR is so effective at detecting tiny genetic signals. Traditional PCR tells scientists whether amplification occurred after the process is complete. Real-time PCR, or qPCR, allows scientists to monitor amplification as it happens. Specialized fluorescent markers generate signals that increase as more DNA is produced. This provides additional information about how much target genetic material may have been present in the original sample. Real-time PCR has become an important tool in both research and diagnostic laboratories. Before PCR, studying small amounts of DNA was much more difficult. The invention of PCR transformed genetics. Many modern advances in biotechnology, molecular biology, and medical diagnostics depend on PCR directly or indirectly. It is one of those scientific innovations that quietly supports countless discoveries while remaining largely invisible to the public. The science behind PCR testing and genetic detection is based on a simple but powerful idea: making enough copies of a specific DNA sequence to detect and study it. Through cycles of heating, cooling, primer binding, and DNA synthesis, PCR amplifies genetic material with remarkable precision. Whether used in disease testing, forensic science, environmental research, or genetics, PCR remains one of the most important tools. Once you understand how it works, it becomes easier to see why this technology revolutionized the way scientists analyze genetic information. Thanks for watching.